I want to start by just showing the basic dihybrid cross. So that dihybrid cross is when you take for your P generation, you take two true breeding, and in this one I just kept the dominants together and the recessive together. When we say true breeding, it basically just means that for each gene, you're homozygous. Now, when we look on the next one, I'm going to flip these so you can see two different ways to do the test cross. But for right now, I just kept them together to make it simple. So you cross these two true breeding pairs, and then what you get is these heterozygous individuals for both genes in the F1 generation. We've done this before, right? And because we've crossed them so they get one dominant and one recessive allele, then the dominant for each one is the phenotype in that F1 generation. But then when we cross those, we get 4 different phenotypes. And in this one, I'm just using the dominant is brown, and then the other dominant is normal wings. Go make the fly one because it's got the easy letters to use. So now when you cross those, I'm not going to do this because I think everybody knows how to do this kind of Punnett square with that dihybrid cross. So good job. But then when you get your phenotypes, when you get both dominant phenotypes, so brown and normal wings, this is normal Mendelian inheritance pattern, which means they're not linked. You're going to get that 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16 ratio. So the 9 to 3 to 3 to 1, where 9 possible phenotypes come out as both dominant, 3 as one dominant, 3 as the other one dominant, and then 1 as both recessive. So if we take 1,000 or we collect 1,000 offspring from this mating of these two F1s that are the same, right? Both heterozygous for both genes. If we take 1,000 of those, then basically in order to calculate this, we take 9 out of 16 divided by 1,000, and that'll give us close to 562. So if our numbers match up to this 9 out of 16, 9 divided by 16 times 1,000, in this one, it's 562, 188, 188, and 62 are a 9 to 3 to 3 to 1 ratio. And this is very close. Now, could you do the chi-squared? So a chi-squared test takes your expected versus your observed and tells you whether or not those are the same statistically. So if you get these numbers, then these are linked. All right, so that's in observed number 1, that would indicate that it's linked. Or sorry, no. Sorry, I'm like getting mixed up again. No, not linked. Good thing I caught that because I would have to do the whole thing over again. These are not linked because they're sorting independently. Now, if you observe 1,000 and you get numbers that are slightly off, so if it's like 62 off here, whatever that math is. So these are going to be, and this is almost half of this one. When you get funny numbers that don't match that 9 to 3 to 3 to 1 ratio, then you have to do something else. It's a clue that these might be linked, right? So then you put now, it's a question mark of whether or not they're linked. Now this dihybrid cross is not going to tell you that. You have to do the next thing. So in this, these are possible numbers that you could get. And if they are more different from this 9 to 3 to 3 to 1 ratio, you have to do the next thing, which is the test cross. And now when we are doing that test cross, I set it up because you can do your test cross in 2 different ways. And I think seeing this in the 2 different ways will help you see how the sorting happens. I also have a red marker this time. Hopefully you can see a little bit. But so you have, when you do your test cross, you do the same thing like you did over here in the P generation, where you take 2 true breeding parentals. And in this one, I kept the dominant ones together and I kept the recessive ones together. So remember, a true breeding within each gene is homozygous, but your true breeding could have a dominant one gene can be dominant and one gene can be fully recessive or homozygous recessive. So you can do either way. And I think doing it this either way will help you see how it can sort differently down here, as why sometimes we get 1 to 0 to 0 to 1, and sometimes we get 0 to 1 to 1 to 0. So your numbers can change depending on what your P generation is. So if you take your P generation and you cross it, you get your F1 generation is always that heterozygous, just like we saw over there. But hopefully what you're seeing now in these, when I have the red a little bit, is that in this one, the dominant ones are both coming from the female, and the recessive are both coming from the male here. And in this one, we have one dominant in the B and one recessive in the N coming from the female, and then one recessive B and one dominant N coming from the male. So the idea is that if these are linked, then these black ones, I'm going to go over here first. So in this one, if they're linked, the black recessive ones are going to move together and the red dominant ones are going to move together. But over here, because we did a different P generation, then the one dominant B and the recessive N are going to move together, and the recessive B and the dominant N are going to move together. So when we look down here in our simplified, because over here we did two heterozygotes, we crossed two heterozygotes together, so we had all these options. But in this one, what we do is we take the other cross in a test cross with those fully recessive, often male. And so that gives us only a recessive option. So that way we trim down our Punnett square into just the 4 options instead of 16 options. And when we do that, we have our possibilities here when the maternal, the red ones here are staying together from that P generation, and when the recessive ones over here are staying together. So on this one, in this particular cross, if the genes are linked, you're going to have a 1 to 0 to 0 to 1 ratio because these are mixed. Now over here, what we're looking for is our big B and our little n to stay together. And so that is this one. And then our little b and our big N to stay together. So coming from this P generation. So what I've starred in each of those is the parental phenotype. So coming from what you have in the P generation, if these are staying together down here, then, oh, and I forgot to do this one as red. Sorry, I'll fix it. There, so now it makes more sense. But so if these are coming down together into this square, into our Punnett square, then those are going to be linked. So now on this one, instead of having a 1 to 0 to 0 to 1, now we have a 0, because that's mixed, to 1 to 1 to 0. So when you're doing that, you can have it either way. And we're always going back to, on this one, my maternal had brown and with wings, and my paternal had black and wingless. That's why it's easier to go back to the phenotype, or, don't go back to what's in here because this is always recessive and it's always just basically you can simplify it by just looking at the gametes from the female that you're getting over here. And so now on this one, if we look back, so now this phenotype is the brown and wingless, which is this female. And this phenotype is the black, this phenotype in the middle is the black, I started that wrong. I was going mixed, right? Sorry. So this phenotype is the black with wings. So you're going back to what you have on these phenotypes, and then you're going to look at your numbers. So I could give you numbers, and I could say, let's do this one since I got more room over here. So if I gave you numbers and I said I crossed 1,000, and I got, I don't know, 490 of this one, 490 of this one, and 10 of this one, and 10 of this one. Right, so now using these numbers, are these linked or not linked? And it is true, we get our 0 to 1 to 1 to 0 ratio, so we know that they're linked. And then the next question is, what percentage of recombination is it? To do that, you do your number recombined over your total times 100. And that'll give you your percentage. So our number recombined is 10 plus 10, which is 20, divided by 1,000, which gives us 2% in the end. So that's our 2%, how you're going to calculate these. If I gave you numbers that were more like this, I'm trying to make them a little bit different, but easy, right? Then that's a 1 to 1 to 1 to 1 ratio, and those would mean that they are not linked. Okay, so now the slightly more complicated problem is if I give you, now I'm going to tell you that you know that these genes are linked. We're going to get 100 offspring. And what we know is that the two genes are 10 linkage map units apart. And the question is, how many expected of each phenotype are you going to get if the genes are linked and 10 linkage map units apart? So in order to do this problem, I've given you the total number of offspring that we're going to do. So you take the math, you take this math problem, and now you say, okay, my total is now 1,000. The number recombined is the thing that we don't know. How many expected would be, what are you going to have in here? So your number recombined is X that you're going to need to solve for. And if we carry out that full equation, then I told you linkage map units, but linkage map units are the same thing as percentages. So we can just linkage map units, 10 linkage map units equals 10%. So we are just going to put 10 here. All right, so now all we have to do is solve for X. And so when we're going to do that, when you have this one, you just divide by 100 on both sides, and to get rid of that. And then you're going to multiply by 1,000 to get rid of that. And that is going to give you your final number. Okay, sorry, I just needed to make sure that we could all see. I didn't know how far I was getting here. I'm barely at the computer. So, oh, yay. Okay. And so when you do that math, you can do it any way that you want. But you can kind of just do it like, okay, I've got that, we cross off that 0, and what I have in the end is 100, right? So, and that means that our number of recombined is going to be 100. You could also simplify it. You can do the math if that works for you. You can also just say, okay, well, if a linkage map unit is percentage, what's 10% of 1,000? You can also do it that way. And then if you wanted to recheck your math, all you would have to do is, okay, if I did it, so I've got 100 divided by 1,000. I've got 100, and I go 1, 2, 3, that's 0.01 times 100. And then you go back to 10, right? So you can work on the math in any way that works best for your brain. So now we have 100 recombined, and so we need to split it between our recombined chromosomes, or our recombined, not chromosomes, but our recombined products here. And so that means that the recombined was, now I have, instead of a big B and a little n, I have a big B and a big N. So now I need to find that one. And I have 100 I have two possibilities for the recombine. So 100 divided by 2 is 50. And then the remaining, I have 900 left. 900 divided by 2 is 450. So that's how you go backwards from it. If you know they're linked and you're given the linkage map units, you have to go backwards with the math. Now, if we come over here, if they are linked, because we did the test cross differently with our P generation, now on this one, our dominants are going to stay together. And so that's going to be the parent one, which is going to have no recombination. So if we look for our recombined ones, we've got the big B and the little n. That's going to be recombined. And then this is going to be recombined, the little b and the big N. Those are your recombined ones. So those are the ones you're splitting across the 100. So now, instead of having it like this, because of the same, same species, or not same species, but same alleles, right? Same genes, but we did the parent generation differently. So we have to think about that. So now the ones that are, when they're staying together, these parent ones are going to be the dominants and the recessive are going to stay together. So hopefully in doing this and seeing it this way, it helps recognize how you can do the test cross if you're keeping the dominant together versus the recessive together, versus splitting them across. You're going to get different numbers or different outcomes down here at the bottom. And that's all I have. So I can go to sleep at night now.